Hello. Good morning, everybody. And first, let me say, thank you so much for the opportunity to give this talk today. I also want to extend my gratitude for the support for my two students, Ariana and Joe, to attend this meeting as well. They're both going to be presenting posters either today or tomorrow. And so I'm actually not going to focus on their work. There's another student, a former student in my lab, Deepti Kannan, who is here. She's currently a graduate student at MIT, but I'm actually going to talk a little bit about the work that she did when she was a student in my lab. I also want to extend my gratitude to the NSF Physics of Living Systems for supporting my work. I very much appreciate it, Creston and Angel. And so let me get right to it. A lot of the talks during these couple of days are going to be focused on the fundamental organization of chromosomes, and really at the heart of this is understanding epigenetic regulation. So of course we have the same DNA in all of our cells, but through the organization of that DNA, it actually expresses the proteins in very different ways, okay? And that's what gives different cells different identities. And so if you look at an EM image of a nucleus, you can see that there are very, compacted regions, these are called heterochromatin, and very open regions, these are called euchromatin. And roughly speaking, those genes that reside in these less densely packaged regions are those that are expressed. Those are the ones that are in euchromatin. And so from my perspective, this is a very physical problem, right? It's physical segregation between, the chromosomal regions, and that actually gives rise to a very important biological consequence, which is how the proteins are expressed. And so from our perspective, we'd like to develop a fundamental understanding of the physical forces that drive the segregation and therefore give the cell its identity, okay? Now, through that, it is necessary for us to sort of acknowledge the fact that the physics at varying length scales is very, there are important consequences at these very different length scales on both the physical organization as well as the biological function. And so what I'm going to try to do today is I'm going to talk a little bit about our work in understanding how nucleosome arrangement affects the local spreading of epigenetic modifications. And then what I'm going to do is I'm going to build to address chromosomal reorganization dynamics. So here we're talking about temporal dynamics and fluctuations of the chromosomal organization. And then I'm actually not going to have any time to talk about this, the chromosomal segregation at very large length scales, more the scale of, say, chromosomes or the whole genome. Okay, but we had beautiful talks today and throughout this meeting on this area, and I'm absolutely interested in talking about this offline, the work that we're doing with regard to large-scale organization. And a lot of our work really complements some of the really beautiful work that's done in Jose's work, lab and Michele and other folks. Okay, so let me first start talking a little bit on a more granular level, okay? A lot of the talks that we're going to see are on, sort of large-scale organization. So here I'm really going to focus more on the nucleosome level and sort of multiple nucleosomes, and I want to understand what is the consequence of nucleosome positioning on the physical organization of the chromosomes. Okay, and so there's been huge advancements in our understanding of chromosomal organization, through experimentation. And one area that has seen some really interesting developments is in the imaging by cryo-EM on the nucleosome organization in living cells, okay, or in fixed cells, but nonetheless imaged in a living cell. And so, historically, the arrangement of, there's all these pictures that you would see in, say, a molecular biology textbook where you kind of, they talk about how there's a fundamental unit, which is the nucleosome, and then it's arranged in something like a 30 nanometer fiber, and then it's sort of hierarchically built up all the way to a chromosome, okay? But if you actually look at the arrangement in a living cell, by these new developments in Claudia O'Shea's lab at Scripps, and you see what it actually looks like, it actually seems that on a nucleosome level that it's more like a zigzag. And it's sort of, a little more random, and it has different linker lengths that are quite, sort of disjointed. And it's perhaps a little bit more disordered than this otherwise very, beautiful fiber that you can get from an in vitro experiment. And so what arises from this is a set of questions of, well, what are the consequences of having this heterogeneity in the linker length on the local organization of the chromosome? Okay, and so here is, the nucleosome core particle, okay? And as you can imagine, if you alter the linker length, even by just one base pair, you're gonna get that it's gonna rotate this nucleosome relative to the other nucleosome, if you have two in a line, quite a bit. And that actually has huge consequences for how the larger scale is going to be impacted by these subtle changes in the spacing, okay? So here what I'm gonna do is, now I'm a theorist. My group does actually quite a bit of analytical theory and some computational simulations. And, from our perspective, we need to be able to just, make a prediction for how these geometric and physical consequences are on a larger scale. And so I'm not going to have a whole bunch of math. This is the only math that I'm going to have in here. And actually, the only thing that I want you to get from this is that what we do is we leverage the tools of polymer physics in order to make predictions about what are the statistical arrangements of these polymer chains, and we can incorporate a great deal of detail, which includes the bending, the twisting, the geometric arrangement of the nucleosomes relative to each other, and we can build that all into an analytical model that is really beautiful because you can make predictions that directly engage experiments, and it's highly computationally tractable. Okay, so we can actually scale from these single nucleosomes, how they're arranged, all the way up to many nucleosomes and predict contacts and all these other things. Okay? So no more math. I'm not going to do any more on that, but nonetheless, that's what we're doing. Okay? So, what is the consequence? If you have a regular array of nucleosomes with the entry and exit angles dictated by the crystal structure of the nucleosome core particle, and you were to dial the linker length ostensibly around what the eukaryotic chromosomal linker length tends to be, which is on average 45 base pairs, you will get an effective Kuhn length. And the effective Kuhn length is essentially a statement of how the random walk at a very large length scale would look. Okay? It's just a matter of what the step size of the polymer on a large scale would be. Now, as you go from 31 through 51, a regular array will sort of go from a compact to a highly elongated to a more compact structure, and the Kuhn length will vary wildly, okay? So in other words, the effective behavior of a polymer chain is highly sensitive to what is the linker length between the nucleosomes if it was dialed slightly, okay? So now, what we can do is you can ask the following. What if we have an average linker length of 41, but we just add plus or minus 2 base pairs or 6 base pairs? Okay? And I'm showing you predictions from both just a geometrical model, which has no thermal fluctuations, and then one where you add fluctuations. And here what you see is something very interesting, is that when the variance goes from 0 to 2, okay, to 4, the effective Kuhn length drops precipitously, and essentially the 0 temperature prediction is the same as that with fluctuations, which is actually kind of interesting if you think about it. What it says is that if you think about the organization on these larger length scales, okay, that in fact, slight variability in the linker length can have a larger consequence than the elasticity of the twist and the bending elasticity of the polymer chains that comprise the linkers alone. Okay, so in other words, geometry plays a huge role in dictating the effective behavior. And in fact, these are snapshots which are just simply geometrically chosen with a slight bit of variance. And you can see that when there's 0 base pair variation, it's a very elongated guy. And just with 2 base pairs or 6 base pairs, it totally crumples up into this sort of ball. So in other words, if you wanted to, you could dial in contacts at a local scale by just subtle variations in the nucleosome positioning that would enable sections that are far distal to suddenly come into contact with one another. Okay? And it's from this perspective that we actually think that nucleosome positioning and variability plays a huge role in these local contact capabilities. Okay. So you can do lots of other things like make predictions for looping probabilities, and we did that. And I'm not going to go into the details of this in the interest of time because I want to get to a couple of other things. And so what I'd like to do is I'd like to talk a little bit about local spreading. Okay. Now, so far we've talked about epigenetic modifications, and we haven't focused on a particular one. So I'm going to focus on one in this talk, which is what is a very common epigenetic mark, which is associated with constitutive heterochromatin, and that is the mark at the tail of the H3 protein of the core histone octamer at the 9th position. So it's a lysine, and that can be mono, di, or trimethylated. And when it is methylated, it actually has preferential binding of a protein, a reader protein called HP1. Okay? And HP1 has a particular characteristic that when it binds to a nucleosome, one of its domains, the chromodomain, binds preferentially to the modified tails. And then the other domain, the chromoshadow domain, is sort of out in solution, and it can oligomerize with other of these HP1 proteins that are locally bound. Now, if you get sufficient binding of HP1 in a region, it will go to a heterochromatic state. In other words, it will condense into this denser region due to the abundance of the local binding of HP1. Okay. Now, a large part of the work that we've done is to understand the writing of that epigenetic code, of understanding how the binding of HP1 will lead to local preference for binding of a writer protein, which will cause local methylation to occur to nucleosomes that are in local proximity to that site, okay? So in other words, the reader, which is HP1, binds to a state which has the epigenetic mark. A writer will then be, I hate the word recruited, but it'll be bound to that HP1, and then it will spread locally to other nucleosomes, okay? Now, actually, it's really interesting. In epigenetics, there are very common players of readers and writers that have common interactions with one another. This is true in this H3K9 case. It's true in H3K27, which is the polycomb motif, and it has similar readers and writers. Excuse me. So there's a beautiful set of experiments done in Jerry Crabtree's lab at Stanford, where what they did was they actually just took the chromoshadow domain of HP1 and chemically linked it to a locus. Okay? And then what they observed is that over a course of 5 days, they had local spreading of the epigenetic marks to the right and to the left of that bound site. Okay? So, very briefly, since we can model the organization of the chromosome at this nucleosome level, what we can do is we can take the contact probabilities associated with specific nucleosome positionings, and then we can say what is the probability of that nucleosome coming in contact with that HP1, which is forced to be at that site. And then through a model which includes the chemical kinetics of the transfer of the methylation and the turnover of the nucleosomes, we can predict the dynamics of the spreading. Okay? To make a long story short, because there's very specific positioning of the nucleosomes near the transcription start site, which happened to be exactly the sequence that Jerry Crabtree had located that HP1 domain, we could actually dial in exactly where the nucleosomes were into our model, which I described in the last couple of slides. And what we do is through our theory, we can predict the local spreading of the marks. Now, the theory is shown here, and in the inset is actually experiments. And what you notice is something really specific, is that the spreading is asymmetric, and it has this subdominant peak to the left and a dominant peak to the right. And the reason for that is very specific. It just happens to be that near the transcription start site, to the left of it, there tends to be a nucleosome-free region. And that has a lower probability of making that loop than the one that happens to be just right next to it. And so therefore, this local organization of the nucleosomes actually has a huge impact in how the methylation marks are spread. Now, a sort of tantalizing result, which we just got, actually Joe just got it, was the following question. Now, there I was just showing you a model where we're looking at spreading of methylation marks near a site that absolutely has an HP1 bound. Okay. So the thing is if you look at the ChIP-seq on methylated nucleosomes, okay, normally, if you think about it, you see a high degree of methylation in the heterochromatin domains and a very low degree of methylation in the euchromatin domains, right? So euchromatin, ostensibly, doesn't really have a lot of methylation, but it has a little. It has on the order of, say, 4 to 12%, okay? And it just looks like noise. But what Joe did is he actually looked at the correlation in the H3K9 in the euchromatin domains, the stuff that just looked like noise, right? And what he found was, statistically significant, is that there is a positive correlation on the order of, say, 18 nucleosomes, which is consistent with the local spreading from Jerry Crabtree's lab. But then there's this secondary peak which happens just around 10 kilobases, and it goes up positive, very small, and then it trails off much lower. So in other words, even in the euchromatic domains, there is a slight coordination of those methylation. And what we think is happening is that essentially the readers and writers are constantly at work, even in the euchromatin domains, where they're just sort of spreading locally, but then it sort of gets damped back down. So in other words, it's always poised to alter its local epigenetic state through this sort of methylation transfer set of reactions. Okay, so I only have a few more minutes. How many minutes do I have? 5 minutes, okay. So here's what I'm gonna do, is I'm gonna talk a little bit about dynamics in the next 5 minutes. And I'm gonna talk largely about sort of larger length scales, but trying to understand how the stress communication in chromosomes actually lead to both spatial and temporal correlation in the dynamics. Okay, so now here's the thing, is that at this sort of length scale, we don't need this high granularity of a model. We need something a little bit more coarse-grained. But on top of it, what we've learned is that we also need to incorporate several additional factors. Some of those factors include environmental viscoelasticity, because the environment in a nucleus is a lot, it's very polymer rich, and the environment's gonna have a lot of elastic content as well as viscous content, as well as active fluctuations. And I'm going to talk very briefly about active fluctuations in just a moment. Okay, so the first observation that was made by us back in 2010, so this is really old work, I just need to point out that because chromosomes are polymers, okay, that they have temporal correlation in their motion. Or in other words, if you have a segment that fluctuates down in this direction at one time, it will tend to stretch out the chromosome, and it will have to recoil in the opposite direction at a later time. Okay, so for example, the consequences of this is that if you look at loci, in this case in bacterial cells, you'll actually see that if you look at the velocity autocorrelation function, it will have a negative peak. So in other words, if you step this direction at one time, okay, that at a later time it will tend to recoil and pull you back in that direction. So this is a very robust signal, and it's been demonstrated both in bacterial cells, in eukaryotic cells, in many different species. And in fact, this may not be so clear on the screen, but these are experimental results the theory is just right on top of the experiments. And so it really shows a robust signal that is dictated by a combination of viscoelasticity and polymer physics. Okay. So, given that I have very little time, I'm not going to be able to go into all of this, but it turns out that because we've developed this model for understanding dynamical rearrangements at these large length scales, we've now been able to extend this to look at the question of if you tracked a whole bunch of spots at one time. Now, experimentally, chances are you don't actually know what their genomic identity is. And so there should be a robust way that we can use theoretical analysis in order to assign what those spots are in a statistically consistent manner. Okay? And so we've developed the analytical theory to do this for the Rouse polymer, and now we're extending this to include the effects of active forces, which are heterogeneously distributed over the chromosome, in order to both assign the genomic position, infer the underlying architecture, as well as to begin to interpret what are the underlying active processes that are happening that drive that chromosome as it's moving around. And so the goal of this then is to allow a framework that experimentalists can use in order for them to look at those dynamics, given the parsimonious nature of how the labels have to be done, and to use that to get both the underlying architecture and the active forces. Okay, so I'm actually going to go through this. I was in more detail than I wanted to. We've confirmed this with experimental comparison. This happened to be on two-spot fluorescently labeled loci in eukaryotic cells. And we've confirmed that the algorithm does indeed give you exactly the accuracy that's dictated from the experiment. And we've developed a whole framework on which you can essentially say what would be the appropriate discretization in time for your acquisition, as well as the positioning on the genome to get the exact accuracy that you would like in order to get the spot assignment and the architecture from those experiments. Now, finally, biological fluctuations. I probably just have 2 minutes right now, right? Yep, I'm done. So here's what I'm going to do. You know, a lot of the work on organization is based on an equilibrium description. And in fact, a lot of the work that we've done on organization in chromosomes is on an equilibrium description. But of course, active fluctuations play a huge role. And in fact, there are many experimental observations, including ones that we've collaborated on with Julie Theriault, that show that indeed the motion of the chromosomes is largely driven both by thermal fluctuations and active forces, okay? So we've developed a polymer model that addresses this, and we've learned something very fundamental. And here's what I'm going to leave you with, is that active forces, because of there is sort of an inherent speed limit on which active forces can be influencing chromosomal dynamics, that speed limit actually dictates that there is a very important length scale dependent consequence of active forces, okay? At very small local length scales, because the time scale of relaxation is so short, it's basically oblivious to the presence of active forces. However, if you look at much larger length scales, large enough length scales where the coordination is on the scale of, say, tens, hundreds, millions of kilobases, or millions of bases, what you're going to find is that the active forces actually elevate the dynamics. And so you get a very subtle but important distinction in the dynamical consequences at these very different length scales. Okay. And I suggest that you take a look at this paper if you want to see sort of the fundamental exposition on this. So I'm going to leave you with that. All right. So these are my conclusions, but I want to leave enough time for discussion. So what I'm going to do is I'm just going to thank my whole group, and specifically I want to point out Deepti, as an undergrad at Stanford actually, did all that fancy schmancy math that was necessary to do the nucleosome positioning stuff and the coordination, and we're doing a number of extensions to that work now. And I'm very grateful to our collaborators, experimental collaborators in particular. And I'm very grateful for funding, and I'm happy to answer any questions.
Super nice talk. So on the first part, I have two questions. So a place in the genome where we see very strong phasing of nucleosomes is around CTCF sites, which according to what you're saying, would mean that at the base of loops, we would tend to see stiffer chromatin. So any intuition of what that would yield? And let me throw in the second question. There's plenty of places in the genome where we expect nucleosomes to be removed by a remodeler to actually pop off a nucleosome. And so here you right away get 147 base pairs extra as your linker. And that would presumably affect a lot this local stiffness. So intuition on what this would give?
Yeah, so your point about CTCF is really good. I haven't thought about that, so I don't have any like speculation on what the consequences for this rigidification would be. It's something to think about. I mean, what we're trying to look at right now is some of those cases where there's sort of a large scale, like there's cases where you have like an immune response where you get very different nucleosome positioning effects, and then contacts would be affected by that. And so that's something that we're exploring right now, something which is more kind of global changes, because then it wouldn't be quite as subtle of a local effect. I'll have to think on the CTCF. And your other point about the position, you know, what we haven't done is we haven't asked the question of what is the time scale of the reorganization of the nucleosomes and how does that affect the dynamics at that local scale? Because all we're looking at is just statistics right now. And so I think that to that other point, there's a lot of really interesting dynamical questions that would arise.
I guess another way to formulate my second question, more in mathematical terms, is if the variability in the spacing is something that has a small but very heavy tail, or you would get a few linkers that are much bigger than your short linkers.
That's interesting. I mean, we could talk about the consequences of that like as if it was a Levy flight process where some of the tails, if they were power law distributed, would lead to very, very large differences in the cell-to-cell variability. I don't have anything more intelligent to say about that right now. I'd love to, we should talk later.
Thanks a lot.
Yeah.
Yeah, fantastic talk. Andy. So I wonder about the first part, similar to what Antoine was asking. If the linkers are dynamically changing, because the DNA is peeling off the nucleosomes all the time, presumably they're going to be very rapid interconversion between all the structures that you showed, right?
So, okay. We did do a version of the model where we included a little bit of peeling and unpeeling and looked at the consequences of that. And it kind of softens a lot of, you know how we had this variability where it's like very big steps. And then this is going to kind of soften those differences, of course. Now, these other questions about like the breathing dynamics and how that affects I mean, it all is sort of a competition of timescales in many regards because, you know, on the one hand, you're talking about like, does the rearrangement happen fast enough on a larger scale that it would actually care whether these local dynamics are happening? I think it's a very rich set of questions. It's something to follow up on for sure.
So, Andy, very nice talk. I really enjoyed the way you put it. I just have a little bit of a question on what I just asked. I don't know what fast and slow is here, so maybe you can tell me what fast and slow means on the time scales that you're rearranging versus the time scale of peeling. Also, how much methylation is important in terms of getting the orientations depending on the size of the link, basically, and how much that just changed the stability that's going to change peeling. So these are two things that come in your model, right? Based on what I saw, you're not getting peeling into the model, you're just getting more how these different linkers can create different organizations.
Context, yeah, exactly.
Right, but how the two phenomena compete with each other.
And how does methylation then affect that local, I can say something very speculative at this point, which I wished if I had COVID again, I would have time to do more research because when I had COVID, I got so much done, it was amazing. And I developed a model. which has to do with the question of how does then the nucleosome positioning and the linker length get affected by the local binding of HP1, and then how does that alter the spreading of those? And we actually have the inklings of what would look like that these sort of very, very slight amounts of methylation could lead to a local linear compaction, and that linear compaction would lead to this sort of something that looks sort of like wiggly noodle sort of things that you see very consistent with Claude O'Shea's work, okay? So the interplay, in other words, what I'm trying to say is that the interplay between the local methylation, even at 10% or sub 10% levels, can lead to local interactions that dramatically alter the organization, quite consistent with that. I don't have anything more intelligent to say just yet. Hopefully this summer we're going to make a lot of progress on that question. But I think that the interplay is really, really important, and it's really cool in the euchromatic regions. The timescale question is, I think, on the one hand, from a polymer physics standpoint, you always sort of think about length scale and time scale being like linearly proportional to one another, or not linearly, but directly proportional to one another. In other words, local dynamical arrangements also have very short timescale relaxation. But I think in this problem, we have to accept the fact that because of biological turnover, it can be very different than that, and motors and other things. So I think that it's not always so simple to just make a statement of basic proportionality in the context of timescale. But there's a lot more philosophical discussion we can have around that.